for interrogating neuronal circuits at high speed and resolution. And that's what I will be mainly talking about tomorrow. So we have developed over the years various types of techniques using optics by which you can record activity of neurons from the brain at single cell resolution at high speed, and also to use optical tools to manipulate neural activity in a living brain. We have also a couple of other techniques, such as super resolution microscopy and optical force spectroscopy, to study interactions of individual molecules with their substrates in biological systems. Today I will be mainly talking about this area that we are also exploring a little bit, and which is what kind of qualitatively new things you can learn by bringing the power of quantum technologies to biology. And one example of that is, as I will be talking about, investigations about the threshold of visual perception. So the question that we have asked is a rather simple one. So we ask, how many photons is it required to elicit the perception of seeing? And of course, we were not the first one to answer this question. There's a history to that question, examining this question that I will be talking about. But to start with, I just want to give you a brief overview of the structure of the mammalian retina. So if you look at how the retina is organized, light enters the eye through the cornea, and then it has first to travel a bunch of cell layers until it hits the photoreceptor cell layers, where light can get absorbed by molecules that are residing in these photoreceptor cell layers, and that will trigger the perception of light. So what you realize first, this is sort of the difference between biology and engineering. So this wouldn't be an architecture that an engineer would be coming up with. Why would you want to have the elements that should be so sensitive to the light putting on the back of the eye, such that the light has to travel through all these cell layer types? But that's basically how evolution came up with that architecture. And so you have in the photoreceptor cell layers two types of photoreceptors: the rod photoreceptors and the cone photoreceptors. So the rod photoreceptors are responsible for perception of light under very dim light conditions, and the cone photoreceptors, of which we have three types, they are more responsible for bright light perception, and they are also responsible for color perception. They synapse then to a different cell layer. They are horizontal cell layers and bipolar cell layers that make transversal connections. And then the other cell layer is the ganglion cell layers, which then which pools basically the information from a range of photoreceptor cell layers and sends it then to the optical nerves, to the visual cortex in the brain. So if we now look at how light perception is elicited in one of these rod photoreceptor layers, that's what we find. So there is a so-called outer segment of the cell that is mainly responsible for collecting the light. If you look here in more detail, you find it is filled with these discs that are embedded in which the rhodopsin protein is embedded. This is the light-absorbing protein. And there's a stack of them just to increase the efficiency of the absorption. So increase the probability of detection of every individual photon that reaches the outer segment. If you look down here in more detail, you find that these rhodopsin molecules, this is basically the protein that is responsible for the perception, has another light-absorbing chromophore that is called the retinal inside. And what happens is that when the retinal is hit by a photon, it undergoes a very fast isomerization reaction. So it goes from an all-trans retinal to a 13-cis retinal. And that isomerization reaction also entails a physical rearrangement of this chromophore that then triggers a range of biochemical reactions within the protein that then triggers the signaling cascade in the photoreceptor cells. So there are two aspects of this process that are really astonishing. One is the extreme fast process of this reaction, which happens on the order of 150 femtoseconds, and it's known to be the fastest biological process overall. And the second is that the extreme efficiency of the system. And so an individual event of absorption of light by one of these retinal molecules is known to trigger a physiological response, a detectable physiological response on the level of a photoreceptor cell, which can be measured electrophysiologically. So going back to the question of now, based on the sensitivity of the system, whether or not humans can see photons, the very first historic experiments in that regard were done in the 40s by Hecht et al., who designed this experiment in which they used subjects who were placed in dark chambers. So they were sitting there for about 40 minutes to get completely dark adapted to the conditions. And then they were looking at a fixation point such that light would be hitting the part of the retina that would be most sensitive to the light perception. So these photoreceptor cells have a different distribution on the retina. And there is an area about 23 degrees off the fovea that has the highest density of the rod cells. So that's why people are asked to look at the fixation point. And then they had a classical light source of which they could control the intensity by using attenuation filters and shutters and so on. And they were sending the light into the subject's eyes. And they were simply asking them whether or not they have perceived the light as they varied the intensity of the stimulus. And from that, they got curves like that, where they had on the x-axis the average photon numbers, and the y-axis the percentages of the flashes that were perceived. And from this, they concluded basically that on the order of 5 to 7 photons would be sufficient on the level of the retina to elicit the perception of seeing, which was defined in their case as the 60% probability of perceiving the light. So these were the first experiments, and they were later on also repeated by other groups in the 70s and in the 80s, but they seem to have some common shortcomings. So first of all, there were a few data points and low statistics. As you have seen in the previous slide, all the conclusions are made only from a few data points here. The second thing is that there were basically no experiments done below a range of 15 photons entering the eye, so no really low photon range experiments. Also, this type of yes-no type psychophysics protocol that I was describing before is known to exhibit some shortcomings. So most importantly, it has this varying false positive response, which makes it difficult to compare and pool data across different subjects. So different subjects, as you can imagine, could have internally different internal thresholds of when they feel comfortable calling something seen or not seen. So it makes it difficult. How do you compare data between subjects? And then most importantly, there's this inherent variability of the input that is related to the Poisson distribution of the photon numbers that you are very familiar with. So what you can do is basically you control the average number of photons, but depending on the number of photons that you send into the eye, you have always the variability related to the standard deviation, to the Poisson statistics associated with it. So at the same time, however, there were then experiments done in the 90s and in the 2000s by Bailey and Rieke, who showed that an individual photoreceptor cell can be very sensitive to individual photons. So what they have done here is that they recorded from an individual photoreceptor cells into a suction electrode kind of configuration in which you can record the current that is generated in a photoreceptor. And when they were shining it with flashes of light, they could see that the responses that they record exhibited a kind of a quantal change. So you have most of the time nothing, then some responses that are at some level around 1 picoamp, and then the next level was changing in a quantal manner. And by looking at the statistics of these, they could in fact see that it would be reflecting the photon number statistic of the input light, which is very interesting. So you can get from this distribution the Poisson distribution of the light, suggesting that on the individual photoreceptor cell layer level, light can be detected with single photon efficiency. So what we thought, yes, please.
So how does this compare to the best, known detectors that people make?
Well, I mean,
Should we just be using those?
well, I mean that is not reliable all the time. So I mean, I would say the most impressive part of this is that this is happening at room temperature, and you are aware the best detectors that we can create require cooling detectors down to low temperatures, and this is not this is just a statistical process, right? It's not really reliable in that manner. So
what's the efficiency?
It's about 60 to 70%. Yeah. Okay, so we thought that we should be revisiting some of these experiments. And so the experiments that I'm going to tell you about were done by two lab members, Jonathan Tinsley, a graduate student in the lab, and Maxim Molotov, a postdoc in the lab. So basically, we set up an arrangement that was very similar to the experiments that I was showing you by Hecht et al. So we had a laser that was controlled by a modulator, and we had the attenuation filters. We fiber coupled this into the dark chamber, and that's where the subject were sitting and looking at the fixation light. We had also a beam splitter so that we could record on each individual trial the average photon numbers that were sent to the subject's eyes, just to control for long-term drifts and any kind of changes that may be happening in there. And so that's sort of the arrangement that you can see. So subjects needed to lean forward, bite on a bite fixation piece in order to keep their focus onto the fixation light. And you have these knobs via which they indicated the responses. So an important aspect of the experiment was also the design of the psychophysics protocol by which subjects were indicating these experiments. So instead of using this yes/no type protocol that I was mentioning before, we decided to go towards an two-alternative force choice protocol, which was done in the following way. So the subjects were sitting there, and whenever they felt comfortable, they triggered just the trial by pressing a button. And after about a second or so, they heard an acoustic sound, and after about 700 milliseconds, a second one. And they knew a priori that coincident with one of them, there would be some amount of photons sent to their eyes, but they didn't know the order, so that was randomized by the computer. So after the second acoustic interval, whenever they felt comfortable, they gave their interval response, whether they felt it was in the first or the second one. And then thereafter, they gave a confidence response, indicating how confident they were in the response. 3 indicating a high confidence rating, and 1 was not, they were not so sure in their response. And afterwards, they also received feedback as to whether or not their responses were correct. So this had a couple of advantages. So the two-alternative force choice is known to remove this subject's specific bias for false positive rates that I was mentioning, because subjects are forced to choose between one or the other. So it's not based on an internal threshold for when they feel they have seen something. And that allows the comparison of the data across different subjects. Also, the rating scheme is known to push subjects to the highest level of sensitivity. And we also incorporated training before the subject entered these experiments to further make them familiarize with the stimulus that they will be receiving. And we use also high statistics. So in total, the data that I'm showing you is based on 30,000 trials. So initially, we repeated the same experiments that I was showing you before. So we changed the intensity of light and looked at the probability of the correct response for various subjects. So here I just want to point your attention to the fact that the baseline is at 50%, because in that scheme you get always 50% of the time, even just by random guessing correct. And then the performance went up to about 90% or so for higher photon numbers. So basically, we could reproduce the results. We get tighter error bars because we had higher number of trials. Next, if we look at the low photon responses, we got a more interesting observation that is shown here. So first of all, we saw that then the probability of the correct response for these low photon numbers were consistently above the random guessing for all of these. But it was not very well fit. We couldn't fit the data very well, but the curve that was fitting the high photon number data, suggesting that here maybe something different, a different mechanism would be going on that I will come to a little bit later. Next, we looked at the distribution of the confidence ratings. So, and what we saw here is that we changed the number of photons from low range to the higher photon range. We saw that the distribution changed from something that was dominated by the low confidence ratings, rating 1 and 2, to higher confidence rating, rating 3, and this was expected, suggesting that at higher strengths of the stimulus, subjects were more confident in their response, so they could actually see the light and give their responses. But I just want to point out this is for if we look at all responses. But if we look at the correct responses, we saw a different phenomenon. We saw, namely, that at the correct responses, they were dominated by the high confidence R3 responses, suggesting that there is a higher probability of the correct response for high confidence ratings, suggesting that this may be the cases where the photons were actually not scattered or absorbed, made it to the eye, causing an isomerization reaction, and that's what subject picked up. So we also then, to further see whether this result would be consistent, we looked at the improvement of the performance as a function of training. So, and we saw a change in the fraction of the correct responses for the 3 confidence ratings as a function of training. So here you see they typically went through about 6 to 10 training sessions. And as they went through the training, we saw that a fraction of the R3 responses picked up and the fraction of the R1 and R2 responses declined, suggesting that subjects got more confident in the responses. They could push themselves to see these weak stimuli. And consistent with that, we could also see a slight change in the probability of the correct response for the R3 responses as a function of session number. So going back to the questions that we started with, what does it tell us? Can humans really detect a single photon? And the second question that we are still left with, what do we make out of this nonlinearity that I showed you at the beginning? So coming from a background in quantum optics, we thought one way how we could answer this question would be eliminating the uncertainty in the photon numbers. So we wanted to use a spontaneous parametric down conversion source to create single photon cases. And so the idea was here basically that we would use a type 1 down conversion process in a non-degenerated fashion, such that we could basically select the signal photon to be right at the wavelength that is the highest sensitivity of the visual perception at 500 nanometer, and ended up with the idler photon being at the correspondingly longer wavelength. So we selected the green part here and detected basically a yellow photon. And so by doing that, we could basically generate effectively a single photon source, and the detection of one photon would herald the arrival of the second photon. So we used the idler basically just as a trigger photon, and then sent the signal photon through the subject's eye. So another question, so this is the source, is the detection. How would we detect that? And here we faced also a trade-off. On one hand, you could imagine that using a SPAD would give you a higher detection sensitivity, as is typically used in quantum experiments. On the other hand, using a single photon sensitive EMCCD camera would allow us to detect multi-photon cases more efficiently, and which we could then reject. So we could operate effectively the down conversion source at a higher pump energies. And there is some trade-off that we needed to look at, since we didn't have access to single photon resolving detectors. So we ran a couple of simulations, and we looked at how the ratio of these multiple to single photon numbers in SPDC depends on the mean number of photons per pulse, both for the SPAD detection and for the EMCCD detection, and found that the EMCCD has a slightly more favorable slope that put us basically in this region, and we basically settled for 0.05 as a mean photon pairs per pump. There was also another trade-off that we needed to look at. Basically, of course, it would be nice to be here as low as possible, but you then pay the price of requiring much higher number of experiments, because most of your cases you end up with empty pulses. So we looked at the required number of experiments as a function of these two detections. And so for this range that I was indicating before, we ended up being somewhere where the number of experiments that we needed to do was also somewhere where it could be realistically done. So that's how we chose then the settings for our experiment. And the setup was very similar to what I was showing you before. So we have just here now replaced the classical light source with the SPDC light source. We have the type 1 down conversion that was, again, fiber coupled and sent to the subject eye. They were looking at the fixation light. And for the idler arm, we had, again, sometimes a SPAD initially, but then during the experiments, an EMCCD detection. And this is the layout of the system, and that's how it was basically realized. So you have your UV light at 261 that goes here and comes to the BBO. You have the green photon sent to the subject eye, and the yellow one being detected by the EMCCD camera. So when we repeated then the experiments that I was showing you before, we saw, and if we looked at all combined ratings across all subjects, we saw that there were the probability of the correct response was still for a single photon above the random chance, including the error bar. But as you can see, the error bar and the probability overall yield a relatively large p-value here. But we saw a completely different picture if we look at the R3 responses again. So for the R3 responses, we had a significantly lower p-value, and this showed us basically that the detection of a single photon with above chance would be possible. In this case, again, suggesting that these are the cases where the photons were not absorbed or scattered by the incoming by the cell layers in the eye, and would actually trigger an isomerization reaction. So we tried to run a couple of tests to convince ourselves that we could rationalize this observation. So the first one was we wanted to see, so how would that compare to the performance of an ideal detector? And in order to do that, we modeled our observation based on signal detection theory, which is just not assuming any threshold, but it's saying, so given two distributions, where is the best place to put a boundary between them. So we assume that we have E photons entering the eye, and which has an efficiency which is labeled with eta, so that combines the absorption of the eye, the isomerization of the eye, and so on. So we get E times eta light-induced signals on the level of the retina. At the same time, we need also to assume a noise distribution, which is just the rate of internal noise of the system, basically random isomerization of the system as it's happening. So whatever is looking downstream of the retina and is supposed to make a decision, is basically its job is to discriminate between these two distributions. So one distribution that is given by D, and the other one that is given by D plus E times eta. So which is the same as asking, so when is the difference distribution larger than 0? Which you can see here. Now this entity is basically has two parameters. One is the D, the rate of the noise-induced isomerization, and the other one is eta. So D is actually very well, has been quantified for primates, and that's the rate per rod cell. However, eta, interestingly, there is a large range of variabilities about that if you look at the literature, so going somewhere between 10 and 30%. So we looked at at how this probability of the correct response for a different range of etas and ds would look like, which are shown in this curve. And the dotted line and the gray arrow bars is basically indicates our experimental result, suggesting that with what we observe is consistent with what you would find within what is known from the literature. So the next thing we did was doing a control experiment. So we had, in order to eliminate any other sources in the experiment that would be probably contributing to this observation, here is basically what we had, what I showed you before, the probability of the correct response for the R3 ratings that we saw when we do this kind of post-selection. Now, because in many of the cases, we end up with empty pulses. Basically, there are cases where there is no idler photon detected in the camera. We know that we can use those cases as catch trials, basically trials in which we know a priori no photon was sent to the subject's eye. So if we look at those cases, we would expect that this should vanish. And this is what we observed. So if we just didn't change anything in the setup, just do a post-selection upon the cases where the camera wasn't detecting anything, we saw that the probability of the response was not to be differentiated from random chance, suggesting that this has to do with the fact that in these cases, the subjects were sent a photon into their eyes. Now, going back to the model that we observed, that I introduced, there is another thing that it would tell you. Namely, it will tell you that this delta M, the discriminability, should linearly increase with E, the number of photons, right? So that is what you would be expecting. But if you remember what I showed you before for low photon detection, we had this nonlinearity in the system, which suggests that this is not. We needed to augment this model, and this wasn't capturing well what was going on in the low photon regime. So to do that, we looked at the probability of the single photon detection as a function of delay between different photons. So remember that the subject could trigger their responses whenever they felt comfortable with. So that allowed us to end up with a situation where we had a range of different time points when they triggered the responses. So we've basically looked at what is the probability of the correct response in a given trial, depending on the delay to the previous trial, right? And when we looked at that, we saw that there was a transient change in the probability of the correct response that peaked around 3 or 4 seconds, and then it went down to the baseline again for longer delays. So this suggested that there is a transient enhancement of the detection probability upon previous absorption of a photon. Now, again, to convince ourselves that this would be real, we then, again, post-selected for cases where the second photon was empty. And when we did that, we didn't see that transient effect. So what this suggests is that the absorption of a single photon leads to a transient increase of the discriminability. And we tried now to model that into our initial model.
What the bottom plot was there?
I'm sorry?
Say again what that bottom plot is.
So that is, again, for the catch trials. And the catch trials are the cases where the idler photon was not detected by the camera. So a priori there is nothing that is going. So that whole transient change wasn't there. So we try to incorporate that now into our model. And so we said the probability of the distribution of signal that drives ultimately the decision variable on the level of humans is given by the probability distribution of noise and the photon-induced events, as I was showing you before. But that times a transient change in the gain. So, and this is sort of indicating that the absorption of a photon will be inducing a transient change in the gain of the system. And basically, the reason why the second photon is detected with a higher probability after a delay is because it is being detected on top of a higher gain situation. Right? So we try to extract now the delay, this gain kernel from a different type of experiments. Basically, we look at the cases where the second photon, now we had a previous isomerization reaction, but in the second trial, it was a catch trial. Basically, we knew that here there was no photon. Right? So the idea was here that you induce a photon that would change the gain, but then you have also the probability of these spontaneous isomerizations that are the dark noise. So if that would be true, then you would expect that the first part of the trial, the first delay period of the trial, would lead to a higher probability of the detection than the second one, and this would basically lead to a relative change in the bias of subjects choosing interval 1 versus interval 2. And this is what we indeed could observe. So from that, we could extract then the gain kernel of the system, and using that, it allowed us to fit both curves, both this delayed detection events and also the low photon probability, the nonlinearity that we observed in the low photon probability case. Yes,
do you know that both photons are being detected by the same rod?
They are in fact not. The probability of that is quite low. But this doesn't need to happen. Yeah. So the photons are distributed in an area that is about 50 to 100 microns. And in there, you would have many rod cells. So with that, in summary, I showed you that we believe humans can detect single photons with statistically above chance probability. So the quantum light source based on spontaneous parametric down conversion allows an effective elimination of these higher photon number states, basically allows us to create sub-Poissonian sources, and that allows us to probe visual pathways in a way that wasn't possible before. So, and at extreme low light conditions, the sensitivity of the visual system temporally seems to be modulated by the absorption of a single photon. So what the real circuit mechanisms are that are facilitating that, and what it means for the biological system, it's something that it's still unclear, and we would like to follow up on that. So some of the outlook and future directions of this is to basically try to understand how is the single photon detection really facilitated, and what is the underlying mechanism of this photon-induced modulation that I was describing. So is it something that is based on the retina, circuitry of the retina, as we believe it's the case? But it could be also a cortex-based mechanism. So it's not inconceivable that this is sort of a modulation of the attentional state. So depending on when the subjects have paid more attention, they could be having an enhanced probability of the detection. And so what we would like to do is to combine some of these experiments with EEG and MEG recordings that allow us to look at the same time at the states of the cortex of these subjects. And what is the nature of this statistical internal noise? So by eliminating the input noise of the system, it allows you to directly access the internal noise of the system, and to better understand what the source of that is and how the biological system facilitates detection on top of the internal noise. So with that, I just want to thank the people who have done the work and were involved. So I mentioned them throughout the talk. So the project was initiated by Jonathan Tinsley and David Wartman, two students in the lab, and was then pushed forward by Maxim Molotov. Robert Prevedel, another postdoc in the lab, was also involved in the design of the light sources and detector simulations, and is currently this project is being pushed forward by our student postdoc Sasha Agne in the lab. Thank you for your attention.